So I thought I'd put this up just for a second here. This is the key you'll find on the Moodle page of my summary. Now you might choose to put more information on here. You might choose to put a little less or pick like your favorite word of, electron carrier maybe, something like that. But this gives you a chance to really summarize everything that I just said in a little too long a time into as few words as possible. And so the second stage as we carry on here is the citric acid cycle or the Krebs cycle. Like I said before, I like Krebs cycle because it's a shorter word, but citric acid is a molecule that's in the cycle, which is also where it gets its name. So the citric acid cycle starts with acetyl-CoA, which shouldn't be too jarring now since we've had that kind of conversion at the end of glycolysis. And generally, it's a series of carbon adding and removing steps. As it removes a carbon in the presence of oxygen, it's going to make CO2, very similar as what we saw from pyruvate becoming acetyl-CoA. And so CO2 is produced here, and it also needs oxygen. So it requires oxygen and it produces CO2. It also produces 2 ATPs and a bunch of electron carriers. If you really want, you can keep track of the number of these and the different kinds, but I think as a good starting point, their role in this is important. So those are molecules that carry electrons to the electron transport chain. So we're going to summarize this, but I thought maybe what we could do is look at this really detailed cycle. So this is straight from your textbook. And really what I want to do on this is just highlight some key things. So the key thing is that acetyl-CoA goes in, and there's 2 carbons going into the cycle. It's going to merge with a 4-carbon molecule, making this molecule right here. So we talked about how pyruvate can be pyruvic acid, or you might remember that carboxyl groups can be carboxylic acids as the functional group on the end of an amino acid. Citrate can also be called citric acid. And that's where this cycle gets its name, is that molecule right there. So if you like to have that context, you could call it the citric acid cycle, or you could call it the Krebs cycle. Okay, the other thing that happens, we'll notice that there's some carbon dioxide leaving the cycle every time we reduce the molecule by 1 carbon, and oxygen is needed to make that carbon dioxide. The other key thing that happens all along here, you can see these molecules, and the schematics here are so nice. Those little blue things are electrons. And so those molecules are literally carrying electrons onto the electron transport chain. Those are those energy intermediates. The last thing to focus here is the product of ATP. Now, I can see that there's 1 molecule of ATP being made here, but if we remember, there were 2 acetyl-CoAs coming into this, 2 acetyl-CoAs when we broke down that glucose molecule. And so this whole Krebs cycle is in duplicate, and that's how we get 2 ATPs out of the Krebs cycle. If I were to summarize this in the key things that really matter is that we have acetyl-CoA coming into it, and it is a series of carbon adding and removing steps in a cycle, right? And so we have oxygen going in and carbon dioxide coming out. The other key thing that we have is these electron carriers or energy intermediates coming out. And then the last one is that we have an ATP molecule coming out. And all of this is in duplicate because we had the 2 pyruvic acids or 2 acetyl-CoAs coming out of glycolysis. And so this doubles the count on our ATP. I don't think that recreating that is really useful. If I were to go to the summary key here now, I can see that this happens in the mitochondria. Yes, it needs oxygen. Acetyl-CoA is going to go in, and what comes out is carbon dioxide, 2 ATPs, and electron carriers. Or you can do, the other naming systems of those electron carriers.